Good afternoon, everyone. Thank you so much for coming. I'm very excited to see you here to join our Amazing Frontiers lecture series. On behalf of everyone, welcome to the Frontiers of Brain Health, where we take a deep dive into the most exciting new developments in brain research. I'm Francesca Philby. I'm a professor in cognition and neuroscience in the School of Behavioral and Brain Sciences. I'm also EP Senior Associate Provost of Faculty Success. And also here at BrainHealth, I lead the neuroimaging of reward dynamics laboratory. So please make sure you stop by and say hello. At the end of our talk, just as a reminder, you will have a few minutes to ask questions. So for the virtual audience, please use the Q&A function. And for those here with us in person, have your questions ready to ask. We'll get to as many as we can at the end. And this presentation is also being recorded, so as a reminder. For those who don't know much about us, the Center for Brain Health is a cognitive neuroscience research center at the University of Texas at Dallas. We have dedicated the past 3 decades to exploring neuroplasticity and the brain's amazing lifelong potential to get stronger and work better. Now, I would like to introduce our wonderful speaker today. I'm so excited. This is Dr. Jiahui Guo. She is an assistant professor of psychology in the School of Behavioral and Brain Sciences and an external fellow at the Center for Vital Longevity at UT Dallas. Before joining UTD, Jiahui completed her PhD in cognitive neuroscience at Dartmouth College with Dr. Bradley Duchesne and did her postdoctoral training also at Dartmouth with Dr. James Haxby and Ida Gobini. Jiahui's research centers on high-level human perception and cognition, exploring the neural and cognitive mechanisms underlying processes like face recognition and social perception in both typical and neuropsychological populations. Her work primarily utilizes naturalistic stimuli and integrates a range of methodologies, including what we know best here at the center, anatomical and functional neuroimaging, behavioral assessments, computational modeling, and artificial neural networks. So without further ado, please join me in welcoming Dr. Guo.
We live in a dynamic and complex world. So in this world, we need high-level perception to help us recognize objects, orientate us in new places, identify people, read their expressions, understand their body languages, and also making social interactions. This organ, the brain, roughly around 3 pounds on average in human adults, protected by the skull, sitting at the top of the spinal cord, is the center or the key for doing all those complex computations. Understand how the brain deals with those complex information is the key for the neuroscientists to understand how the central nervous system works in real life. So in this talk, my key question is, how does the brain enable high-level perception in real life? Previous studies have already provided us with a bunch of regions. The regions are, in the back of the brain and ventral temporal cortex. Those regions, they specifically responsive to a certain type of stimuli. For example, here, you can see the red region. It's called FFA on the ventral temporal cortex, and responsive exclusively to phases. It's not exclusively, but very sensitive to phases. Okay, and those regions are not the only regions which can be responsive to a certain type of stimuli. There are other regions, for example, the regions which are responsive, very sensitive to other categories like bodies, places, characters, or objects. Those regions are also not only just clustered in the back of the brain. They extend to the anterior part of the temporal lobe and also in the frontal lobe. And all those regions across the brain form an extended network for the high-level perception. Okay, but after we learn where probably those computations can take place, do we already know how the brain enables high-level perception in real life? The answer is no, because those regions are mainly defined in the normal population or typical population. That's the central part of the population in this distribution. However, we have those people who are at the tail of the distributions. Understanding how their brain works is not only can help us to design suitable education or healthcare programs for them, but also provide us with great opportunities to understand how the brain works from the diverse perspectives. So it's important to study these neuropsychological populations. And the second, we see those regions, they basically are at the same locations in individual brains. However, in real life, that's not the case. There's a lot of individual differences going on in the location, the size, and the shape of those regions. So for example, here are two individual brains, and I'm plotting the FFA, the fields from phase area in those two individuals. And you can see the regions, they are wildly different in those two brains. Which means in real life, in real analysis, we need to find a method to deal with these idiosyncratic topographies. And the third part is the regions. The regions I just showed in the last slide only told us where the computations may take place, but didn't tell us how the information are represented in our brain. In other words, we need to understand the representations of those naturalistic information. So we need to focus on the representations and not only the locations. So in my study and in my research and my talk today, I will have three parts. The first one is I'm going to introduce the research in neuropsychological populations with a group of people who have difficulties recognizing faces. And in the second part, I will introduce you with a novel method which deals with the idiosyncratic topographies with naturalistic stimuli. And in the third part, I will discuss the representation of dynamic faces using naturalistic stimuli and deep convolutional neural networks. So let's now focus on the first part. And we see faces every day, and the faces are important source of information for us to, deal with the world. Because we need to recognize the people, recognize the face in order to communicate with them, know what's the situation and complete different tasks. For our normal population, for our typical participants, it's easy, it's effortless for us to recognize faces. It just happens instantly. However, for those special population who have difficulties recognizing faces, this is a really challenging task for them. And we call this group prosopagnosia. And there are two types of prosopagnosia existing. First one, we call them acquired prosopagnosia, or AP. And like the brain we show here, those participants, a lot of them, they have those lesions in the brain, either due to any disease, epilepsy, or brain tumor, or some accidents. They lost a part of the brain, and they acquired this problem later in their life. And another type of prosopagnosia is called developmental prosopagnosia. The definition is they have face identity impairments in absence of brain damage or broad neurodevelopmental problems. And here are two examples of personal experience let you be more familiar with how those people experience in everyday life. Okay, what's their experience with faces? One is I listened to an NPR episode recently and realized that other people didn't have to use shoes to recognize their friends. And the second one is I once asked a friend to introduce me to the person with whom she was speaking and who turned out to be my sister. So these people, they really had difficulty in recognizing faces, even with their relatives or their family members. And those difficulties often have substantial effects on the quality of their life because they cannot recognize faces. It's going to be really problematic in their daily life. And the roughly 2% of the population having this problem. That's roughly 6 million Americans and 130 million people worldwide suffer from this problem. In the past several decades, the central question of DP is trying to understand the neurobasis of DP. Here I'm showing you the classic face-active regions on the typical inflated brain. And those regions can be divided into the anterior areas and posterior areas. There are two competing theories telling us what kind of problem are going on in the DP's brain. One theory is called disconnection syndrome. People who are holding this idea thinks those people, DPs, they have normal posterior areas, but the white matter tracks connecting the posterior areas and the anterior areas are impaired, which leads to the deficits in the anterior temporal cortex. So the problem are just in the anterior part of the brain. The other idea is called distributed account. People who have this idea, they think the DPs, they have deficits in both anterior and posterior areas. There's evidence supporting this idea, which includes people with DPs were found they have reduced physical activity and functional connectivity in posterior areas, and also structural abnormalities in the vicinity of the posterior areas were also reported in the previous research. And the people also found abnormal N170 in their brain. And we know there's potential of the N170 coming from the posterior part of the brain. Previous studies actually suffers a lot from several different types of disadvantages. The first one is most of the DP studies, they only have a small sample size, which has insufficient power. The samples, because those populations are very difficult to get, so people only include probably even fewer than 10 people in their study. And most of them focus on a few regions in the brain, which had only limited coverage of the face system. And nearly all of the studies, they used static face images, which is highly constrained in the ecological validity of their studies. And the last point is they used a classic threshold-based method, which they hold the same threshold for both the normal work control participants and the DP participants. And because we know the DPs often have weaker responses in fMRI in those regions, so they cannot even be localized with this method. And those people which cannot be localized will be excluded from their analysis, which just in turn contribute even more to the smaller sample sizes. So we need some new method to improve this or correct this bias estimation. On the other hand, in our new study, we try to include as many DPs as possible at that time. So we had 22 DPs and 27 controls in our study. This number doesn't really seem very big, but it's one of the largest DP studies in the literature already. And we try to probe the entire face network. So we had a full coverage of the face system in our study. And we use the dynamic phase videos in our research, trying to improve the ecological validity of our design. And we invented a new method. We call that variable window method. And this method, basically, we don't have a hard exact the same threshold for the DP and our typical participant groups. Instead, we have a mask and we choose the top X% most category selective voxels or vertices from those masks as the region of interest we're going to analyze later on. And this method is much more objective and also will help with the future replications. So the first step, the first question we asked was, because we know those DPs, they have severe problem recognizing faces. Considering their impaired face recognition ability, we were curious whether they still have the similar general organization in their brain of category selective regions. So here we plot the peak coordinates of those face selective regions on its inflated brain. And the darker shades stands for the control participants, and the lighter shades they stand for the DP participants. And we can see for all of the categories, the peak coordinates, for the two groups, they are very similar for all of those regions, which suggests although they suffer from severe behavioral problems, they still have the same general architecture of the high-level perceptual network. And the next question is we go into detail to see whether the category selectivities are still preserved or similar in DPs compared to our control participants. So here I'm using red fusiform face area, red FFA, as an example, and defined the face selectivity as the difference between the percentage of the response to faces and objects. And we can see the face selectivity is significantly impaired in the DP group. This could be either the DPs had a higher response to objects or because they had a lower response to faces. To see which is the answer, we split the responses to the faces and objects in both of the groups. And we find actually, the DPs, they had comparable responses to objects, but had a significantly decreased activation to faces in red FFA. The results I just showed you came from the top 10% most face selective vertices in the red FFA region. We also changed the percentage we used for this analysis, and we can see for all of the percentages, which means we are changing the ROI size, and we found our results are still preserved for all the different kind of percentage numbers we used, which suggests DPs had a pretty stable decreased activation to faces in red FFA. This is also not only true for red FFA, we also plot the results for all the other face selective regions in the network, and we found DPs had widespread face activation impairments across all the regions. And we can directly test the two competing theories, the disconnection syndrome and also the distributed account. So we averaged their face activity in the posterior and anterior areas, and we found for both posterior and anterior regions, DP had impaired face activity. So these results didn't support this disconnection syndrome because they think DPs only have problems in the anterior regions, and on the other hand, supported our idea of this distributed account. From the previous slides, we know there's other regions which are category selective. So not only for faces. So we are curious whether DPs are, they are also impaired for other category selectivity in the other regions. So we tested this hypothesis in the body selective regions and in the place selective regions. So we found DPs, they also are impaired in the body selective regions and the place selective regions, which suggests DPs, they have widespread category selectivity impairments across all of those high-level perception network. So in summary, in this part of research, we found DPs have normal general architecture for high-level perception despite of their impaired behavioral performance. However, their selectivity are actually impaired. The selectivity are reduced broadly in face selective areas, and the reduction spread out to other category selective areas in DP. All those results suggested the face recognition deficits in DPs are only the most noticeable visual recognition deficit. So in the future, our neuropsychologists should widen their lens to look at other deficits in their research. In this part of study, when we are trying to localize those category selective regions, we used a classic scan sequence called localizer. This type of scans, they will ask the participants to watch different kinds of category information in the scanner, recording their brain responses in block design. And for example, in this study, we used the localizer called visual category localizer. So we'll see the stimuli for different categories. If you would like to localize other regions, for example, you want to derive this retina topic mapping, you will have to run another study, run another localizer in order to get those regions. So the classic localizers, they are really useful, they are classic, and they are very precious, they are valuable. However, they are still have some disadvantages. First one is the stimuli, the localizers, they are impoverished and specific, so you will need different or separate kinds of localizers for separate topographies. It's not very efficient. If you are interested in different kinds of regions, for example, the regions which are sensitive to calculation, the regions which are selective to language or other type of regions or theory of mind regions, you'll have to run a long study trying to incorporate all of those different kinds of localizers into our scan. And the second is look at this. They are just checkerboards. They are very, like experiment, like in-lab design, which also lacks ecological validity in the design. And the third one is, this is not very natural, not very pleasant. So you probably have already experienced this in the scanner. So it will be difficult for the participants to maintain attention for those tasks in the scanner. And of course, it's not friendly to those special groups like kids or patient group. So we need something new in order to improve those. So this is a naturalistic stimuli. Very different from those localizers. Those naturalistic stimuli, they have rich information and it can activate multiple brain systems in parallel. So when you run just one movie, and you can probably derive a lot of brain regions from just one scan. And the second thing is the movies, they have better ecological validity compared to the classic localizer design. And you just lie down there, you don't have to even do a fixation, you can enjoy your time in the scanner. So it's engaging and have a very naturalistic experience in the scanner. And it's much more friendly to the special groups, especially for the infants and the special patient groups. So the question is, how can we derive those regions or derive the regions we would like to study using naturalistic stimuli? So in the second part, I will introduce a method which can use naturalistic stimuli to derive the regions. So in this study, our aim is trying to predict the category selective regions or other regions from a test participant using just the movie data of those participants. In order to predict this, we will need the data from an independent group of participants. We call them training participants who have both the movie data and the traditional local ladder data. The method we used to do this is called response hyperalignment. So I'll just plot the data here. And if we take each vertex as a dimension here, so their response to the movies are actually in those high dimensional space. And if we do a rotation or transformation using the response hyperalignment method, which is basically based on the Procrustes, that method in linear algebra, so we can rotate the response in the high dimensional space for each training participant to the space of the test participant. So after doing this, we will get a transformation matrix for each training participant individually. And then we can apply this transformation matrix to their traditional local ladder data for each of the training participants. And then after this step, all of this, their local ladder data will be in the space of our test participant. And then we will do a traditional general linear model and averaging across the participants and the rounds to get the map we call the hyperalignment predicted map. And if we don't do this transformation and we don't do anything fancy, we just do a traditional analysis that's just basically apply the general linear model to the local ladder data and also do this averaging, we can get this surface alignment predicted map. So I'm going to use the face active map, the face versus all the other categories as my example here. This is the ventral temporal cortex of one example participant. And to view the results more clearly, I will zoom in the smaller part to give you a better view. I just plot the results we got using their own local ladder. If we don't do anything, it will look like this. So now I'm going to show you the results we got from the prediction. So the prediction can be done by either using the hyperalignment or don't use hyperalignment, just use the traditional surface alignment. And if we use our method, we can see the predict map looked nearly the same to the map we got from their traditional local ladder. However, if we don't do the hyperalignment, and we just use this traditional surface anatomical alignment, and we can see the prediction is blurry without any specific or participant-specific information. This is not only true for one particular participant, this is true for all of the participants. That's another participant I'm going to show you here. Then we correlated the predict map using hyperalignment to their own local ladder data, local ladder map, and also correlated the map we got from the surface aligned prediction to their own local ladder map. And then we averaged those correlations to get these stats. So by using hyperalignment, we can see we improved the correlation significantly. So the results or the performance we got using our hyperalignment method is significantly better than using just the surface alignment. So we compared our prediction to the noise ceiling, which is the upper bound of our estimation in theory. And we can see our prediction is very close to the noise ceiling already, which means our prediction is really good. We also did this replicated this in another data set, and we found the same result. And we plot the results for every individual in this analysis for the two data sets. And the x-axis is the performance we got using the traditional anatomical alignment. And the y-axis is the results we got using our hyperalignment method. And we can see for each of the individual, using our method will improve their performance. But this method has some, disadvantage because we require every participant watch the same movie. But this is now going to be true in real life because those data could be collected in different institutes. and people are going to speak different languages, and they could be collected from different scanners, and even using the same scanner, the parameters could be very different across different studies. And the researchers often have their own freedom to edit and choose the part of the movie they would like to play in the scanner. So the data could never be exactly the same across different studies. So we need a method to generalize across those diverse situations. So the next part, I will introduce you, the new method. In this study, we want to predict the test participants' category selective topography using just the movie data. And we need, similarly, another group of independent participants who have both movie and localizer data. But this time, the movies could be different from like either the content, language, scanner, parameters, etc. So they could be, the top part could be totally different. And this method we use is called connectivity hyperelement. So you will see a very similar picture here, but different from last time. This time, we need to calculate their connectivity profile first. Basically, that's the correlation between each nodes in a region to each target vertex on the other part of the brain. So here, you can see on the left panel, each line of those checkerboards, it's not checkerboard, those color boards. The color stands for the correlation between each cortical nodes in a region to the target seat or the other vertex in the other part of the brain. So we build the connectivity profile first, and their connectivity profiles are in those high-dimensional space. And similarly, we will rotate the high-dimensional space to best match those in our target part, in our test participant for each training participants. So we will get those individualized transformation matrices again. And then similarly, we will apply these transformation matrices to the localizer data for each training participant. We'll get their transformed localizer data in the test participant's space. And then we will, in this real study, because we want to improve our performance, we did iterations for multiple times. This is a little bit more complicated. If you have questions, we can discuss later. So then we, similarly, we did this GLM and averaging, and by doing these transformations, we'll get this, we call the connectivity hyperelement predicted map. So that's CHA predicted map. And if we don't do anything, just run the traditional analysis, we will get this surface alignment predicted map. So that's SA. Again, I'm going to show you first whether using this new method, we can roughly replicate the results we got using the same movie. That's similar to the last step. So here, that's the results we got from their own localizer, from the own localizer of one particular participant using the phase selective map. So that's still phase versus all the other categories. And I'm going to show you here the CHA predicted map. Very similarly to our last result, the prediction using our method looked similarly or nearly exactly the same to the map we got using their own localizer. If we don't use our method, just use the traditional anatomical alignment, we can see the results are just blurry and no specific individual specific information. So we replicate our first result, and the most interesting part comes is whether using a different movie, whether can we get similar prediction here. So I'm going to show you here, this is the prediction we got using a totally different movie. So that's called cross-movie connectivity hyperelement prediction. We can see no matter whether you use the same movie, the prediction are very similar across all the three, which means our method can generalize across the contents. We plot each individual for this between and within prediction performance, and we can see whether you use the same movie doesn't matter. And this is not only true for one category, we generalize to different categories and they remain stable. And we average the values across individuals and get this group prediction. And the gray bar, or the black bar, is the noise ceiling, that's the upper bound. And the gray bar are the anatomical predicted performance, and the pink bars, the lighter one, is the within movie prediction. And the darker pink bar is the cross movie prediction. And we can see both cross and within movie predictions, they are significantly better than the anatomical alignment prediction and very close to the upper bound, or the noise ceiling of this study, which means our predictions are really good. And this is also true for all the other categories. And we replicate this using several different data sets, and we found similar results. So this study told us we found a method which can predict individualized topographies with high fidelity across diverse situations. So in the second part, summaries, we first found the individualized topographies can be estimated with high fidelity using naturalistic stimuli. And the second is using a optimized the new method. We can we can generalize across different features, different parameters, and different contents to predict those maps. And our method can be used to integrate those high-level cognitive functions across datasets from the laboratory worldwide. Imagine you have your own data collected in the lab, and you are doing analysis with those public datasets somewhere else, but they don't have the regions you really are interested in. You can use our method to use their data and our method and get the regions you need from the in-lab smaller datasets you collected probably years ago. So we discussed first the results we got using a special population, and we introduced the novel method with naturalistic stimuli. And the third part is we're going to discuss the representations of the dynamic phases using deep convolutional neural network. So we see real phases every day. Those phases, unlike those static images, they move their heads, they look around, they laugh, they smile, and they cry. Those phases communicate with us through those changeable aspects of features. For example, expression, eye gaze, and head orientations. Previous studies understanding these representations are focusing on the static images. They offer us with valuable information, but they cannot answer us how those representations are made in those dynamic or naturalistic situations. To overcome this knowledge gap, we developed a database. We call that hyperphase. It's a high-dimensional phase space to examine or cover as much variations as possible in our phase space. This dataset involves a large number of dynamic phases, and here are two example stimuli in this dataset. And currently, this hyperphase dataset includes 707 naturalistic video clips of familiar faces, and each video clip is 4 seconds long. And they vary widely in the identity, age, ethnicity, perceived gender, head orientation, etc. And it's one of the largest naturalistic phase stimuli set in the literature, and we also took those faces into the scanner and collected their brain responses. And this study is a multimodal study, and we use the multimodal approach. And we know from current development of artificial intelligence, and those deep neural networks, they can match or even exceed human level performance. So we first took our data set and let it go through the phase trained deep nets, and we will get the embedding vectors from each layer of this network. And then using this embedding vector, we can build a matrix, which is called representational dissimilarity matrix. So for this matrix, each cell in this matrix stands for the dissimilarity between each pair of the video clips in our stimulus set. And for the cognitive part, this is a deep neural network part. for the human cognitive part, we asked our participants to do a behavioral task. We call that arrangement task. So participants will see the faces, the video clips around this circle, and their task was to arrange those faces based on their facial appearance. So they will do, they will just drag the faces into the circle and do the task. And this is the results from one example participant. Although this is from one particular participant, but interestingly, their results look very similar across individuals. So let's take a closer peek into how they cluster those faces. So they're gonna first cluster faces to two clusters based on the perceived gender of the face. So the female faces are on the left for this case, and the male faces on the right. And for the other dimension, they will cluster faces, based on the age of the face. So you can see those young faces, they are more to this dimension, and older faces are to this dimension. So they will just also cluster faces based on the age, and they also cluster the face based on the skin color of the faces. So the darker shades come into the inner side of the circle, and the lighter shades to the outer side of the circle. So the participants, they miraculously all did the clustering for roughly similar patterns. And for this task, after we got this result, we can build a matrix. We call that behavioral RDM, using the physical distance between each pair of the video clips. And for the human neural part, we took the signal set into the scanner and recorded their brain responses while they watched those video clips. And then we will get representations from each smaller part of the brain. We call that searchlight, but that matter. And you got these live numbers from the small part of the brain, and build another representational dissimilarity matrix based on the distance or dissimilarity of the brain representation across each pair of the video clips. So then we will get all the three different types of RDMs. And we asked two key questions. The first one is, do deep convolutional neural networks, the human face clustering behavior, and the brain, do they share similar internal representations? Which here we are talking about understanding the relationships between each pair of the two types of RDMs. And the second question is, if we find some similarities or dissimilarities, what are the underlying mechanisms for those? First, let's move on to this, the convolutional neural network and the behavioral. We correlate these two types of RDMs for each layer of the face deep neural networks we studied. And we can see the correlation was low in the beginning and gradually going up and then going down. And the peak correlation was very close to the noise ceiling we got estimated from our behavioral study, which means the deep neural networks actually captured a lot of the meaningful variance in our behavioral performance. And this is also true for the other two face networks we used in this study. To understand why the deep nets can capture our behavioral performance, we asked another independent group of participants to do this feature rating task. They need to label the faces either as male or female, tell us the age range, the ethnicity, the gender expression, and the overall head orientation. And after we got those labels, we created an index which reflects the distance in the difference in the day-score, spatial distance of the between and within feature groups. So let's use this as an example. Each dot here is a video clip, and we can see very similar to our behavioral performance. They cluster the female faces on the left, male faces on the right. And this index stands for how clear the divisions are between the clusters. So the higher the index is, the clearer the division will be between different clusters. And then we plot this index for all the features we got for the behavioral task. And we can see our behavioral tasks were mainly driven by the perceived gender, age, and ethnicity, which is consistent with the eyeballing of the data. So they are the most important features we use in our human performance. And we also did this, plot the index for every layer of our deep net, and we can see for the three most important features, the pattern is going up and going down, which means the deep net gradually captured information and squeezed them out. And this pattern was basically similar to the pattern we got for the correlations, which means the deep nets and our human behavior, they were driven by the similar features. The deep nets didn't really capture the expressions much, and the heterogeneous had a very different pattern there. So the decisions and our behavioral representations, they capture the similar features. Then we tested the correlation between the deep nets and our human brain, and we did this for each small part of the brain and go across the entire cortex to get a map. And then we did a multiple comparison analysis, and we found the regions left covered well with the regions in the phase-selective network. So we averaged the correlation in the phase-selective and the non-phase-selective regions, and we found for each layer of the deep nets, they showed like the phase-selective regions captured or had a higher correlation than the non-phase-selective regions. And this is also true for the other two deep nets. But the correlation, the average correlation is very small. It's less than smaller than 0.1, which means less than 1% of the meaningful variance was explained by deep nets. You might ask, probably it's because our data is crappy. It's crappy, but actually this is not true. We calculated this noise ceiling for the whole brain, and we found the regions, especially the phase-selective regions, where the regions we care about, had a really high reliability. It's nearly over 0.7, which means we had reliable neural representations in our data set. And the smaller correlation was because there's a lot of rich information in the neural representations. The last part is the behavioral and the human neural RDMs. We also did this for the whole brain, and the regions which had the highest correlation also overlaps with phase-selective regions. And we did this for the phase-selective and the non-phase-selective regions, and we see there's still significantly higher correlation in the phase-selective regions. However, this correlation is also small, which means this type of behavior also only represented a part of the rich information in our neural representations. And then we plot the index for the neural region of interest in our phase-active network. And the left part is the left regions, and the right part is the regions in the right hemisphere. And we used this still perceived gender as our example here. And we found this information is better represented in the right hemisphere ventrotemporal cortex, which is consistent with our prior knowledge that the static phase features are better represented in the ventral regions than the dorsal regions. And we also using this, the MDS plot to show this. And we can see there's some kind of division here, but the division is not as good as our behavioral performance or the best layer of our deep net, which means there's some representations of this information in our brain, but this is not all of the information. We also did this for the other features, and consistent with our knowledge, like the static features like age, they are better represented in the right hemisphere ventrotemporal cortex. And the dynamic features like expression, they were better represented in the dorsal regions of the right hemisphere, which is also represent or consistent with our prior knowledge that the dynamic phase features are better represented in the dorsal pathway of the phase network, which means our brain actually captured where those information are actually part of our rich information in the neural representations, but only a small part of that, because you can see the index here is very small. So we can go back to our two questions. First, we found the representations of the phase decision and the behavioral clustering task. They captured similar major categorical phase features like the perceived age, gender, and ethnicity. And second, we found the phase decisions and the behavioral clustering task. They modeled only a portion of the variance in the rich information of the neural representations of naturalistic phases. And third, the neural representations of naturalistic faces are rich and containing information beyond those categorical features. So in the future, like if we want to make the artificial intelligence more like human brain, we probably need to take in the dynamic information and designing more ecologically valid objective functions in order to make those networks perform similar like the human brain. Okay, let's go back to the central part of our question we discussed in our talk. That's the high-level perception in real life. So we used the special population called prosopagnostics to understand, their face responses in the face network and other category selective networks. And in the second part, I introduced a novel method which can predict the individualized topographies with high fidelity using naturalistic stimuli. And the third part, I introduced the representations of naturalistic stimuli using deep convolutional neural networks and our behavioral performance. Hopefully, I convinced you today that the human high-level perception is complex and information-rich. And I have been actively developing new methods in our lab and trying to understand how the brain deals with this high-dimensional information in real life. And I would like to give my thanks to all of the collaborators and the funding agencies, and also special thanks to my lab too, and I'm recruiting, so if you are interested, please contact me. And thank you so much for your attention, and thanks so much for the invitation.
Thanks, Jiahui, for an amazing talk, walking us through how your work stemmed to lead us to the multidimensional type of research you're doing. We do have a few minutes for questions, if anyone has questions for Dr. Guo.
That was a great presentation. For the face blindness, is that just like your face, or you said that people recognize shoes? Is that like people's feet, or is it just like, does someone have to wear the same shoes every time, or is it like the aesthetic of the person?
Well, that's an interesting question. For those DPs, they have specific impairments to just the one category, like the faces. They cannot really recognize faces, but they can use other features. For example, the hairstyle, their clothes, their shoes, like their body movements to help them recognize faces in real life. But they have a specific impairment to face recognition.
Thank you. Yeah, fantastic presentation, super interesting stuff. What's an example of how we could utilize this understanding of high-level human perception in AI?
So let me rephrase your question. So you ask me how we can use AI to understand human high-level perception?
Yes, and also what like a functionality of understanding human perception at that level of AI could be. Like visual tracking or just stuff like that. I'm curious, like the future of AI using these models and using this understanding.
Okay, I think for the current AI, well, this is still something we debate a lot in the field, like whether we really need to make the AI similar to the human brain in order to let them finish some tasks. So, or whether this is also something we discuss in class, whether we need to have those, AGI, those more like generalized intelligence. So still it's now no, we don't know whether we need that. But if we wanna use AI to understand the brain, or wanna make the AI look like the brain, we need to first use, not only static images, we need to at least use naturalistic stimuli to train the model. And also we need to, use more, we call that ecologically valid objective functions. So for example, they need to not only tell the identity of the face, this is how the face networks were trained for now, but you probably need to, identify the person or more, in high dimensional space. And also probably designing more other cognitive functions. For example, for a lot of those face networks, they don't have the module like memory or attention like human brain to allocate the resource or use the archived data in order to help them, have a better representation of the entire person. So there are all possible ways we can develop in the future for the AI to be more like human brain for high-level perception, or just use the AI as a tool to understand the brain.
I'm going to cheat and ask two questions. The first is, I know from my own mom's experience that facial recognition is one of the leading indicators of succumbing to Alzheimer's. Is there any correlation between what you're seeing in how you map this and what they see in Alzheimer's patients? Secondarily, what do you hope to do with this knowledge? In other words, you're doing a good job mapping and predicting the why. Is there any hope with neuroplasticity that any of these things can be mitigated or modulated?
Well, thank you for the questions. I think for the first one, whether the face recognition ability relates to Alzheimer's disease, we don't have the data to prove for that, but this is something I read in the literature. Yes, that's true, because face recognition ability is one of the important part of the general intelligence. And people who have Alzheimer's disease often show more severe impairment of their face recognition ability over the years. So this could be an indicator, but we need more data to really have a rigorous answer to that question. But personally, from my own personal perspective, I think yes. The second question is, after we can predict those regions, how can we use the knowledge to better understand human high-level perception? Predicting the regions is just the beginning of the research. First, we need to generalize the prediction to different kind of populations. The prediction we got now is just using the typical population. So actually, in our lab, we are testing whether this method can be generalized to DPs and other clinical populations. That's the first thing. And if we can do this in the different, different populations, we can use the regions we predicted to understand how the representations and the computations are changing in those special populations, and whether that could be related to their clinical performance. And if so, and whether we can design any programs to improve that and solve the problem.
I think we have time for one more, and then we'll go to the chat. You go.
So I have a question about the hyper functional connectivity. So I guess the assumption is like you assume between subjects they have similar pattern of functional connectivity. Is that?
Well, that's more technical, detailed question. So the first thing, the method is called connectivity hyperalignment. Why it's called hyperalignment? That's because we will not functionally align different brains together in a high-dimensional space. So this is a hyperalignment. While this method, we don't actually assume anything. We basically calculate their functional profiles or functional connectivity profiles. So their functional connectivity profiles will be in a high-dimensional space, because you can imagine each connection could be one dimension. And then our tasks, we just use the computational method to find the best match between the test participant and our training participant to derive the transformation which can complete this task. So after we get the transformation matrix that describes the relationships between the two brains in the high-dimensional space, and then we can apply those transformation matrix to the other data to make sure all the other data are in the other functional space, the high-dimensional space we need to work with. So this is more like the logic of the analysis. So we don't assume their functional connectivity profiles are the same.
Thanks, Jiahui. I think we will answer one question from our virtual audience. I'll just choose one. Has your data indicated any subjective emotional factors that influence facial recognition?
Yeah, that's a good question too. I think from our data, we found like the emotion or expression recognition, the identity recognition, and also the face perception. They have, they are in different dimensions, which means people can, those DPs, they have severe problem recognizing faces, but they can still read the facial expressions. So that's very interesting. And also, yeah, they cannot recognize faces, but if you give them two faces, they can perceive this difference between the two faces. So we think the face is more for identity recognition. It's more, we call that holistic information. It's the whole, it's more important than the adding of the parts.
Thank you so much. Please thank Dr. Gold for the wonderful presentation. Thank you. Lifelong Brain Health, this is our shared mission. Thank you, Dr. Gold. And please come back next week. We look forward to seeing you on November 15th when we're hosting Dr. Noah Offman. She's a professor of psychology at the Center for Vital Longevity at UT Dallas. She directs the Cognitive and Brain Development Laboratory and will discuss her research on the development of memory systems in the brain using intracranial recordings. Thank you so much for coming. See you then. And take the Great Brain Gain Text Challenge. There's the QR code. Thanks again.